So it's my distinct pleasure to introduce Mike Kobor, who is a professor in the Department of Medicine, Department of Medical Genetics at UBC, and senior scientist at the Center for Molecular Medicine and Therapeutics. He holds the Canada Research Chair in Social Epigenetics and serves as the director of the program on social epigenetics at the Human Early Learning Partnership HELP at UBC, which was founded and established by the late Clyde Hertzman, who made a big impact on understanding early life and well-being. His research program is focused on illuminating the developmental origins of health and disease across the human lifespan. And I think that my enthusiasm is perhaps a bit aggressively enthusiastic, although someone has arrived. I noticed behind me. And so hopefully we'll fix this. And I wanted to mention just a little bit in the interest of the fact that there it is, yay. That epigenetics, as being part of our talk and having Mike here, is really one of the major innovations of today for a lot of us. So I personally am really excited about the fact that genetic and genomes being mapped as part of CLSA and other studies. So I look forward to hearing what Mike has to say, and I'll let him go before the projector dies.
All right. Fingers crossed that we get through the next two presentations. So it's a real pleasure to be here, and I want to express my gratitude to Larry and the organizing team for having me, inviting me to be part of this exciting 2 days. It really speaks to the foresight and the vision that Larry and others, and also the Canadian Longitudinal Study of Aging, have to include epigenetics in the discourse here, because it's actually, despite how much you might have heard about it in the popular press, it's actually quite a new field, especially as it relates to population epigenetics and aging. So what I'd like to do today is to discuss with you work that we have been doing to map out the epigenome in relationship to human aging, with the premise to understand how environments affect health and behavior across the lifespan. So I'm first going to spend some time to briefly introduce this idea of epigenetics, what it is, and why it is emerging as a key factor between the interface between the genome and the environment. I will then show you some data that suggests that epigenetic variance in an adult population in part actually reflects the biological embedding of early life experiences, but also current lifestyle and psychosocial factors. We'll then move on to talk about DNA methylation signatures of aging, starting with the idea that there might be different signatures depending where we look at. I will introduce to you this really fascinating concept of the epigenetic clock that might act differently, that might tick differently than the chronological clock, and show you some early data from a community cohort. And lastly, talk about environment and epigenetics, again using a interventional cohort that we just started to be engaged with. And so the definition of epigenetics you can see here. As you might have already heard, I grew up in Germany, and Germans are supposed to love definitions. I actually don't, which is probably explains why I'm in Canada. But for those of you who do. So the particular field, as Larry mentioned, that we are really engaged in is to take this concept into the social world and really try to understand the process by which life experiences influence the way our genes are expressed and shaped. And what we're basically looking at is the idea that experiences affect biological pathways, which in turn affect all the way down to the level of the gene expression. Of course, this can also be viewed as a reciprocal pathway. So really, I know that Susan and others always mention this idea of from cell to society. Really what I think we are doing, we're going from society all the way to cell. And so this idea really has flourished in the last few years. And what really underlies the activity of gene regulation is shown here. So I want you to forget everything that you ever heard of genes, okay, because it's a complicated concept, and just think of light bulbs. Okay? And think of a gene as a light bulb. And a gene can obviously, a light bulb can be totally off or totally on or anywhere in between. Okay? And so that's, for the purpose of this presentation, what I'd like for you to think of. So we roughly in our body have 25,000 light bulbs. We have 25,000 genes. So this is somewhat simplistic, but so what the epigenome then is doing, the epigenome actually serves as a dimmer switch for the light bulb. Okay, and so this is shown here. So we can have the dimmer all the way down, we have the light bulb off, we can have it all the way up with the light bulb on. So basically, what the epigenome does, then it regulates the activity of the genes. So for the purpose of this talk, we're going to talk primarily about the dimmer switches, but we're also going to talk to some extent about the light bulbs. And this really, I think, hopefully helps you to not get flustered about genetics and all these kind of fancy words and DNA structure. It's really that simple. And so basically, one of the key components of this epigenetic machinery, that dimmer switch machinery, is a very simple methylation group. Who remembers chemistry? Methylation group? What is it? That's right. Very simple chemistry, as simple as it gets almost. So basically what methylation is, this methyl group can be attached to the DNA itself. So you can see here the DNA strand. And in this particular case, when we have no methyl, the dimmer would be all the way up, the gene would be fully expressed, the light bulb would be totally on. Now, watch what happens. We get the first methyl group. There's a little methyl group coming that's attached here, pushes the dimmer down. Another one pushes the dimmer further down, and look what happens to the light bulb. Till eventually we push the dimmer all the way down and turn the light bulb off. So that's really all you need to know about epigenetics. Seriously. Now, the one complication that we're going to touch upon a little bit on this talk is that if we take our human body, we have many, many different cell types. So we have two I've shown here. Any idea what those two are? A neuron, very good. And that ball-looking thing? That's a white blood cell. So I hope there's nobody in the room who thinks that these two cells look alike. Otherwise, we have to send you to an optometrist. So these cells look very different. They do very different things. And the reason they do very different things is that these light bulbs, actually, if you were to use a microscope and look into the light bulbs, you could see 25,000 light bulbs, and here we have 25 of those, so only 0.1%, that have different intensities, all the way on, all the way off. And so the programming of these light bulbs during development of the human body by these epigenetics marks, by the dimmer switches, leads to the fact that we have these different cell types that look very different and, of course, do very different things. And as you know, unless you apply some heavy genetic engineering, you can't change a neuron into a blood cell or vice versa. Now, the reason why epigenetic really has come to the forefront of so many people's attention, especially in the interdisciplinary research area, is that it has become clear over the last few years that on top of this programming of the developmental epigenome, what can happen is that the environment can tinker with this. And so, for example, look what happens when we have a certain environmental exposure. You can see in this particular case, in both cases, in both cell types, the light bulb got brighter. We can also think of another exposure where, in this case, the light bulb got brighter here, and here was turned off. And so basically what this means is that we can have an environmental programming of the epigenome that acts on top of the developmental programming, but can be specific to the tissue that we look at. And that one speaker this morning mentioned about brain versus central and peripheral. We come back to this because this is obviously one of the biggest challenges that we face if we want to do population epigenetics, such as in the CLSA, and want to derive principles that act upon in the brain, for example. Now, to me, I like visual things. To me, this is still the very best example of epigenetics. So what you can see here are 5 young mice. And again, I think we all agree that this mouse looks very different from this mouse, correct? So if you see that kind of picture, what would you assume, what do you think spontaneously, what underlies these differences in their coat color? Different genes, right? Like they might have gotten different genes from their mom and their dad. Like I have dark hair, my son has blonde hair. Well, what I'm going to tell you is that these mice are genetically identical. They were bred in the laboratory by some, person in the basement in the mouse facility to be clones of each other. There's no difference in their genetic makeup. But what's different in these mice is that actually the circuitry between light bulb and dimmer switch is altered. So in the yellow mice, you have the light bulb fully on and the dimmer switch all the way up. Now, see what happens. If we go over here, the dimmer switch is pushed down, the light bulb is turned off. Interestingly, as you can see on my slide here, what does affect the circuitry between the dimmer switch and the light bulb is actually the nutrition of the mother during pregnancy. Various food components have shown to be able to affect that balance of dimmer switch and light bulb. So that's really interesting. But of course, this is just after birth, and it really affects only, so you think, the color of the mice. And I would bet that very few in the room here care about mouse coat color. No? But what happens with these mice? So let's see what happens when these mice grow up. Let's focus on the yellow versus the brown mouse. Any ideas? Are they going to maintain their coat color? Yes. But watch that they do. But look what happens. So I don't think I need to explain very much, but you can see the yellow mouse is huge. Brown mouse is lean and fit. And this is at the sort of adult, the equivalent of human adult age. So what happened is that somehow the nutrition of the mother during pregnancy has set that dimmer switch in a particular way that's persisting all the way into adulthood to create dramatically different health outcomes. These big mice develop all kinds of problems, such as metabolic disease, diabetes, you name it, solely dependent on what the mom ate during pregnancy. Now, again, this is an experiment done in some basement in a mouse facility. Is there any relevance for human health? Well, there is. So many of you will be familiar with the Dutch famine at the end of World War II when the Germans occupied parts of Netherlands. There was a scarcity of food, and women lived on less than 800 calories a day, but nevertheless, many of them got pregnant. And there are numerous studies about the Dutch famine, but one in particular, published in 2005 in PNS, showed that if you look at the white blood cells of 60-year-old people that were born during the Dutch famine, as compared to people in neighboring areas where the population didn't starve, what the researchers found is that there was a difference in their dimmer switch setting 60 years after they were born in a human population. And it wasn't just a random dimmer switch setting, it was a dimmer switch setting that's involved in growth and insulin biology. So very similar to the data that I just talked to you about from the mouse. There are numerous studies now that show that prenatal smoking actually also can affect these dimmer switches in children and adolescents. There is really seminal work from McGill University, Michael Meany and colleagues, to speak about the fact that the quality of the maternal care, so the social environment, can affect the dimmer switches in rodents and in humans all the way into adulthood. And I'll show you, no, I'm not going to show you some related data to this. And lastly, as you well know, identical twins are often used for genetic to understand genetic cause of disease. What actually turns out that if you look at identical twins, who are probably the closest we have in human populations to those mice, that they actually often acquire even fairly genetic diseases at different times in their life with different severities. And most importantly, a work from Manuel Estella and Art Petronis has shown quite convincingly that their epigenome diverges over time. So if you look at the epigenome of monozygotic twins early in life, it's fairly identical. If you look at monozygotic twins at 60 years of life, their epigenome in parts might quite diverge. And this principle is underlying this idea of the epigenetic drift that will, in aging, that will come back in a few minutes. So there's really a wonderful article in the Time magazine a few years ago, Why Your DNA Isn't Your Destiny. It talks about the potential for epigenetics from an epidemiological perspective, really one of the best reads even until today. What we within our lab and within the CNSA at large, really the model that we propose to move forward is really to take epigenetics together with the environment, but also, of course, in the context of our genetic variation, as an integrative model, how to explain phenotypic variation in a developmental context. And we've done numerous number of works, published numerous papers that speak to this and support this. Most importantly, I think we have shown and others that consistent with this epigenetics contributing to phenotypic variation, there's actually, if you look at a human population, there's actually substantial variation in the epigenome among all of us. And so when we first started this, we really tried to understand, well, what caused the variation in an adult cohort. And we focused at a time, we're still focusing, on the idea that these early life experiences get under the skin to change, to sculpt these dimmer switches in a way that lasts all the way into adulthood. And so that in part, epigenetic variation in adults can be predicted, for example, by their early life socioeconomic status or the early life parental stress level. This is part of a large body of research that we're doing together with Greg Miller and Edith Chen, who used to be at UBC and now at Northwestern, Tom Boyce, who used to be at UBC, now at UCSF. You see his theme here. Speaking of Vancouver as an attractive place. And Marilyn Essex at the University of Wisconsin, and most importantly, as Larry referred to, the late Clyde Hertzman, who really was a champion of this kind of research, and many of you probably knew Clyde very well. And so what collectively we have shown is that indeed epigenetics can mediate these effects of early life environments. What motivated us in part are studies such as these that you probably are well familiar with. The famous Johns Hopkins physician study that showed that at the age of 55, there is a more than threefold increased risk of cumulative probability of coronary heart disease in more than 1,000 white male graduates from Johns Hopkins Medical School that can be traced back directly to whether they grew up in low childhood socioeconomic status or in high socioeconomic status, with, of course, the folks who grew up in low socioeconomic status having a higher risk. Again, consistent with the idea that early life environments get under skin to shape our adult health. And so Clyde has put forward this idea of the biological embedding of early life experiences to really have profound effect on health and behavior in adulthood. We started to do numerous studies to test this mechanistically and with the hypothesis that epigenetics, so the dimmer switches and the light bulbs, are indeed underlying this kind of perpetual effects of the early life environment. And so in a study that was published in PNAS some years ago with Greg and Edith, we could indeed show in a cohort of adults, healthy adults from the lower mainland, that were stratified by their early life SES, so low versus high SES. We could show that this figure here, that the dimmer switches in their white blood cells were actually different. And again, they weren't different in a random fashion. They were different in a fashion that suggested that in the people that grew up in low early life SES, there was a higher propensity for the immune system to overreact. And indeed, when we then take their white blood cells and we challenge them with agents that are commonly used to that the immune system would be exposed to, we could show that indeed the immune cells from the folks with lower SES do have an increased response, a higher response to these agents. Physiologically, we could show that the folks who grew up in lower SES have a higher output of cortisol during the day, so they're somewhat more stressed all the time. And lastly, in a more recent paper, this is p-value distributions, we could show that the early life SES indeed is associated also not only with the light bulbs, but also with the dimmer switches as judged by the skewed p-value distribution. So it basically matters on the level of the light bulbs, on the dimmer switches, the functional response of the immune system, and the physiological response to stress, whether you grow up in this kind of dwelling versus this kind of dwelling. Interestingly, there is no association of the current socioeconomic status, so how wealthy or not you are in adulthood, with the dimmer switches. And that's we can discuss why that might be. We've published subsequent papers to show that some of these detrimental effects can be buffered or by interventions such as increased maternal care. We also found that in adults, there are demographic factors such as gender and ethnicity that are associated with the dimmer switches. Perhaps more interestingly for us, we also found that in adults, their total cortisol level as well as their perceived stress level is associated with levels of DNA methylation in white blood cells. Now, having shown you that, hopefully shared with you why the epigenetics at the interface of the genome and environment, and discussed very briefly some of the epigenetic variants and its predictors in adults, I want to switch to go to more an aging focus. And this came about work we have been doing with Michael Meany from McGill University of Gustavo Turecki. As you probably already could tell, a lot of our focus really is on mental health, on behavior and such. And because we have these light bulbs between the neuronal cell and the white blood cell that are so different, the question that everybody in the field has, well, what can we really learn from looking at the dimmer switch setting in a white blood cell or in a cheek swab, which is the material that we can commonly get, about what's going on in the brain. And so Gustavo, as some of you might know, directs the Quebec Suicide Brain Bank. So he has lots of brains that are from people that died. Some of them suicide, some of them of natural causes. And he also, in some cases, had white blood cells. So we set out to ask, what's the discordance and the concordance between those tissues? Without going into too much detail, when we do principal component analysis, we found that the majority of the variants could be accounted for whether we look at brain versus blood. Then we could also show that some of the variants could be accounted for by the actual cellular composition of the brain. The brain is, as you know, not homogeneous. There are glia and neurons and other cells in there, or the cellular composition of the blood. So that was somewhat illuminating, but was actually more interesting for us is when we looked at other variables that would explain some of the variants. What we found is that the fourth and the fifth principal components, so a fairly substantial amount of the variants, could actually be explained by the age of these subjects. And interestingly enough, principal component 4, and I should mention this is a cohort of only 20 people because, as you can imagine, it's not that easy to get these brains. Principal component 4 was correlated with aging, so these are young to old people, and these are the different, this is principal component 4. Principal component 4 was explained by age in brain only, not in blood. Principal component 5, in contrast, was explained by age both in brain and blood. So that was very surprising for us because up until then, everybody thought that there's perhaps one signature of aging. Our data suggests that there are two, even in the same tissue. Sometimes good things happen when you least expect it. So as we're kind of scratching our head why this could be, a large epidemiological study was published in Cell that actually used blood cells from 700 people to look at the relationship between the dimmer switches, the DNA methylation, and aging. And we used those data to basically try to replicate what we have found in our small cohort. And that's shown here. So what we basically did, we projected these principal components onto the data set of the 656 individuals that were published in Cell. And long and behold, what you can see is there's no correlation with the PC4, that's aging in brain only, but it's a very good correlation of 0.55, and the p-value is 10 to the minus 51 with this when we project this principal component onto blood for PC5. So this suggests that indeed there are two signatures of aging, one in the human brain and blood. And it can be replicated in a much larger cohort. So that's very exciting for us, and we're currently writing this up for submission. Now, of course, in the context of a study like the CLSA or some of the studies that were touched upon this morning, it's impossible to really look at brains. We are switching gears a little to make our life just a bit easier and really starting to look at this idea of the epigenetic clock in a community cohort and environment and epigenetics in more controlled settings. Some of you might have seen the work of Steve Horvath from UCLA, who has done really fantastic work bringing up this idea of the epigenetic clock that perhaps might tick in all of us in different tissues, so related to what I just showed you. It was featured in the New York Times and in Nature and such, really fascinating work. And so basically, what the epigenetic clock, the way we like to look at, and this is currently under review, is that if you have an age-associated dimmer switch, where, say, the dimmer goes up with the methylation goes up with age, it really could be due to two different reasons. It could be due to epigenetic drift, like I briefly touched upon in the monozygotic twins. So this is our subject A. The green one happens to be the monozygotic twin. You can see that there is a difference in their dimmer-switch setting. But if you look at a population, it kind of goes all over. So it's more individual-specific changes that occur during the lifetime that might be stochastic, that might be due to environmental influences on the individual level, but they're really not suitable to look at a population level at this idea of epigenetic aging. In contrast, the epigenetic clock that Steve Howard proposed, and that I'll show you in a second, is actually really likely to be true, is then that in that population, and for example, the two twins, all of their the methylation sites change in the same way. So perhaps that's a broader property of the epigenome in a population, and really suitable then to look at a population level at this idea that the epigenetic age might be different than the chronological age. And so what we have proposed then is basically in the context of health that perhaps the epigenetic age predict the future health trajectory. And so if we were to compare the chronological age to the epigenetic age, as ascertained by the Harvard epigenetic clock algorithm, we would think that the majority of people would be in a concordant zone, but there might be some folks that are epigenetically old, quote unquote, or epigenetically young. And that would go hand in hand with relative health, where you can see the folks that are epigenetically old would have lower levels of relative health compared to the concordant or the epigenetically young folks. And so then the question becomes, now, is this really true if we look at a population, say, for example, the CLSA? Hopefully we'll do that in the CLSA soon, but luckily we have a lot of collaborations, and so we started out looking at this in a cohort, an NIH-funded cohort with Greg Miller and Edith Chen, where we have white blood cells from 600 people across the broad age range. And what you can see here is brand new data, where we basically put the Harvard epigenetic clock to test. So what we did, Steve Howard on his webpage has a algorithm where you submit the methylation profiles of individuals. So we did this for our 600 individuals, and then it spews out the predicted epigenetic age. And then, of course, you can compare it to the chronological age. And so what you can see, red is female, blue is male. By and large, there's a pretty good concordance. And we arbitrarily chose this as a concordant zone. So this is very early days. We need to sort of think more what really is the concordant zone, because you can definitely see there is quite a bit of spread of up to 5 years on each side away from the perfect correlation line. But really, there's certainly outliers. So there's certainly some folks like these guys here that are epigenetically old. Like, for example, if you look at this person, their epigenetic age would be something like 70, and their chronological age is like 42. So you see a huge discrepancy in some of these cases. But you see also the opposite, where this person's age, epigenetic age, would be 40 versus their chronological age is 70. So then that, of course, really, first of all, I think it really beautifully shows that if we look at this, use this principle of the epigenetic clock in a community cohort, we can see lots of concordance, but also really interesting discordance. And then the question becomes, is this predictive of worse health outcomes, like I was hypothesizing? Is this predictive of better health outcomes? Is this perhaps correlated to disadvantaged early life environments and such? So you can ask all kinds of questions with this kind of approach. And we hope to use, as Susan alluded to, we hope to use this same approach in the CLSA with 10,000 people. My last slide I want to show, this is, again, a brand new study that I felt the data is preliminary, so the details of it might change, but we're very excited about it. This is the Walk of Life study that some of you might heard of, that Maureen Ash and Martin Dawes here at UBC have started, and they invited us to be a part of it. It's basically a senior intervention study, where I think 60 old age home people that live in old age homes are encouraged. It's a randomized control trial to undergo activities similar to what we heard this morning. And Martin and Maureen have agreed that we can take white blood cells before and after the intervention. I should say we just really finished the collection of the data, so we haven't really analyzed the intervention per se yet. But what I want to show with you, again, this is principal component analysis, is to look at some of the more obvious variables and whether they map onto any of the principal components that explain patterns in methylation. So you can see here this first principal component, for example, explains roughly 25% of the variance in methylation, and you can see it's associated with weight and BMI. Not surprisingly, perhaps, because some of you might have seen the Lancet paper recently that spoke about a correlation between BMI and DNA methylation. There are other really interesting variables that we heard about this morning. So, for example, the percentage sedentary behavior is associated, I believe, if I see that correctly, with the second principal component. The physical activity and step counts are associated with various principal components. So again, collectively suggesting that the environment that we live in, the environment that perhaps is created for us, and is reflected in the dimmer switches. And so, really excited about this work, and hopefully can discuss later this afternoon how similar approaches could be included in some of the studies that were presented this morning. So, just want to finish by basically summarizing that hopefully I have shared with you some of my enthusiasm why I believe the epigenetics is really at the interface of the genome and the environment. We talked about that some of the variants in adults reflects biological embedding of early life experiences, such social environments, such as socioeconomic status. Didn't really talk to you much about the data we have on family environment, or a study we do with Harvard folks at Harvard School of Public Health on early life abuse. So collectively suggesting that it's really a vestige of the social environment. But we should also be aware, of course, that current lifestyle factors such as psychosocial and demographic factors correlate with dimmer switch setting in adults. We talked about this issue that we're all facing when we want to do population epigenetics, about the signatures in peripheral versus central. And I showed you that at least for aging, which we're all interested in, there are complex patterns that might occur across tissues or be confined to one tissue. I briefly introduced to you this idea that Steve Howard has put forward of the epigenetic clock and how we are putting it to test in a community cohort to show that there is indeed concordance and discordance of epigenetic and chronological age. And the question, of course, that becomes then, is this a vestige of the lifestyle or the environment or a predictor of health outcomes? Lastly, I introduced to you very briefly the walk of life study, where we look at the sort of intervention, the environment in a cohort of seniors, and have preliminary data that suggests that sedentary behavior, weight, physical activity predict epigenetic variation. And what we, of course, ultimately would want to use this for is, among other things, to assess and stratify potential intervention strategies. So I just want to acknowledge all the people who have done the work. These are the folks in my lab that have really led the various studies, all graduate students, postdocs, or technicians, and current and previously. I think I highlighted all the key collaborators that have been part of this journey. And I just want to say that we really have been benefited tremendously from the Canadian longitudinal study of aging, not only because of the technology that we have been putting to this task, but also conceptually to the ideas and that have been brought forward by the CLSA. And lastly, this has been funded by a variety of organizations, including the NIH and the CIHR and the Canadian Institute for Advanced Research and Brain Canada. Thanks a lot.
Do we have time for some public questions?
Yes, please.
First of all, thank you. That was excellent. I mean, to engage everybody right after lunch with something on DNA methylation is really masterful.
Thank you.
That was one of the best discussions of this that I've heard.
Thank you.
And I'm from Stanford University, where they talk about this stuff a lot. So your last slide was really intriguing because you talked about stratification of intervention strategies. So I'm an interventionist.
Right.
We're always interested in trying to target things, but I'm trying to figure out what the argument would be to have to go drill down to DNA when, in fact, you're talking about obesity and physical activity and all of the sort of behavioral markers?
Yeah, so I think part of what, and we have some data from an intervention cohort with David Olds, the Nurse Family Partnership, which is the obviously at a very young end of life. Part of it is, as you probably know much better than I, that not everybody in a given intervention responds the same. And so I think what we're envisioning, and again, this hasn't really been put to the test rigorously in many different interventions, that perhaps having the epigenome could predict whether a given person responds better or worse to an intervention, and perhaps guide some alternative strategies for individuals. That's where I think we see that. Because we don't, I mean, of course, the most obvious would be to find a biological basis for why overall an intervention works or not, or some guidance. So, for example, as I showed you with the poverty, we identified overreaction of the immune system, so that you could think of some therapies that would, potentially, or interventions that would target certain parts of the immune response based on this. But I think really the gold mine that we've put forward, certainly in the context of mental health, would be stratifying the individual response.
That makes a lot of sense to me when the interventions are biological.
Right.
It makes a little less sense to me when the interventions are behavioral / environmental / social / fill in the blank. Anyway,
yeah.
Now I better understand what you're talking about.
Yeah, sorry for not making that clear enough.
Yeah.
If we have more time, maybe we can discuss it later.
Yeah.
Larry.
I really, I feel confused by, what do I say? I a lot of directions to go with it, and maybe Abby said that more eloquently. But what I'm wondering is methylation, if in fact the degree, so you show that different rats or mice that are born, their genome is the same genetically. It's the methyl.
Exactly.
So that was that's a separation from the genetic material effectively, and then how it's expressed is what we see.
Yes.
So the twins make that also clear. So if that's true, and then early life sets the clock, not the clock, but sets the way the mice look, for example, that expression, then if methylation occurs as a result, if it's locked in, well, I guess it's a question, is it locked in, or is the expression, is methylation is occurring throughout life?
So it's a great question.
Due to your exposure, is that happening? Because I thought that was...
It's a great question, and up until a few years ago, it was supposed to be entirely locked in. And so, for example, that's why if you look at the 25,000 light bulbs between a blood cell and a brain cell, they're quite different, and dimmer switches would be quite different. What has become clear over the last 5 to 7 years is that it can be affected by the environment to some extent. Of course, much less. You will never, by, for example, being exposed to a rough childhood or drinking lots of beer, you will never make a blood cell a brain cell. That's not going to happen. So it's more that what I try to highlight. It's more subtle changes. But some of them can be reversed, yes. And most traumatically, that of course is known. So there's an entire field of epigenetics that I didn't even touch upon, which is cancer epigenetics, where of course you see black and white changes, not the kind of subtle changes that make a mouse look yellow versus brown, but wholesale changes. And there are actually drugs that can reverse some of those. There are also natural compounds, such as, for example, bisphenol A that you might be familiar with, that can tweak with these epigenetic markers. But a key question is, say, that really nobody's been able to answer. Say you're in a given environment that changes a given dimmer switch. You're then taken out of this environment. How long is this persisting in a human population? Because what really hasn't happened, and now is happening thanks to CLSA, is that we don't have repeated collection of DNA. Because most DNA, that even the ones that we have been using until the very recently, most DNA has been collected once because it was supposed to be used for genetic variation studies. And so you can, derive certain principles and interesting findings from this. But of course, what you really would want to have is the longitudinal aspect of it. And so CLSA is now for the first time putting us in this position that we can actually look at this, correlated with different environments, exposures, and of course on the other side, health outcomes, and then bring it together.
Thank you, that was very helpful. And just really quick, is methylation inherently bad or can it be good?
It can be both, it can be anything, yeah. Yeah, sorry, I should have made that more clear. It can be bad and good. And just as I said, keep that in mind. It's only one of approximately 150 different epigenetic modifications. Don't want to confuse it too much, but it's the one certainly in a population setting that's really, at this point, the only one that's being used to interrogate the epigenome. Yes, please.
Very interesting. I just was wondering how strong do you think the environmental exposure would have to be? Like, I'm just thinking of an example of, say, a kid grows up in a wooded neighborhood versus a concrete neighborhood. And even if you held physical activity constant, could just the exposure, that kind of exposure, have this effect of changing the gene expression? Or how subtle could it be, I guess?
I think that's an excellent question. And again, that's absolutely nothing is known about this. What is the severity or the extent that you have to be exposed to a given environment? But it could also be, does it matter whether it's, long exposure versus short exposures? Those kind of things are really not known. And I think I'm really excited because I got probably another 25 years in my career. So there's a lot to do. And this is certainly one of the things that we're very interested in, the timing and the extent to which these things or do, for example, do similar exposures cause similar changes in the dimmer switches, or, are the different exposures, say violence versus nutrition, do they act on different substrates? All those kind of things are not known, but are great questions.
Does this mean Lamarck was right?
There is certainly a fraction of epigeneticists that are big fans of, that are trying to bring back Lamarckian thinking. And for those of you who know them, we had up at the hospital, we have a mini-med school once a year, and I had the pleasure of being the dean of the mini-med school some years ago. And Ron Barr, who some of you might know, and Tom Boyce basically staged a play where Tom, I think, was Darwin and Ron was Lamarck. And we were supposed to film it, but somehow the person didn't actually film it. So unfortunately, there's no history of it other than the oral history, but it was great.
Do you need to move on?
I was just going to
take one more.
actually just a comment on that. I mean, it seems to me that while these observations may be, in a sense, new in the sense of we're discovering them, they've been happening to us forever, if it's real. And so in that sense, we've all experienced, as has everybody before us, these very changes.
Yeah, I think what this now brings to the table, and I'm sure you had these discussions with Clyde, is really a biological underpinning that we, for example, hope that if we think ahead about policy and things, that naively we think that if there is a biological substrate for some of these things that, as you said, have happened to all of us over the history of humankind, it might help to enforce the need for policy changes and things like that. I should say that we have one paper that is going out next week with the Pasteur that is just fascinating. I didn't want to show it because the guy at Pasteur is paranoid that somebody will steal the data. It's going out to Nature. But it basically looks at pygmies and Bantus in Africa in an urban versus a rural, in the urban versus the forest setting. And the data is just mind-boggling. So, of course. So, thank you.
Okay, so our next speaker is Elizabeth Christensen. Betsy's professor of psychology at the Center for Research and Education on Community Resource Services at the University of Ottawa. She's a health measurement expert whose research focuses on contextual determinants of health, and she's also a co-applicant or a co-organizer of this particular event. So,
thank you. So I'm going to switch gears a little bit and talk about a systematic review of area effects on health. And it's kind of like bringing coals to Newcastle, I feel, telling this group of people about research on the built environment and health. But anyway, you'll notice that I have preliminary results. So I'm happy to hear if you see a study missing, if you find anything that you don't agree with. I'm happy to hear it because it is just preliminary. Okay, so I'll talk about the background and objectives just a little bit because we want to get to the meat. The methods a little bit, the results. I'll give a general overview of included studies, and then I'll give a results for our 4 areas: chronic disease and mortality, functional limitations, health behaviors, and mental health and cognition. Okay, we've heard a lot about healthy aging and why it's important, but one thing I want to emphasize is how important older people are to their community. They make the largest portion of people who care for grandchildren and other people. They are huge volunteers. They do less volunteer work on average, but they spend more hours, and they're often the pillar stone of the community. And I was telling Susan I came across a fascinating little stat this morning when I was searching through it. In the UK, a volunteerism agency has done a study, an economic costing of the contributions that older people make versus what they cost the system. And the contributions are something like 173 billion pounds, whereas the cost of the system are only 135 billion pounds. So they're a huge net asset to their communities. And because of that, there's this wonderful emphasis on making communities more age-friendly, and this, as Susan said, this amazing conceptual model of the WHO. And but to do that, we really need evidence-based interventions. We want to know why things are happening, what mechanism, and what will work. So we need to understand the aspects of the built environment and how they impact the health of older people, and how they impact on, and how they, the mechanisms through which they act. And I should point out this wonderful gentleman. The pictures I have here are from a photo voice project that I ran a few years ago. And we got 125 people from across Ottawa to tell us what they saw about their neighborhood through pictures. And we had a group of 30 seniors in various communities. And this was our oldest participant, and his name was Marcel. And I hope he's still alive. But he had the idea that he was going to be pictured taking... He was going to have someone take pictures of him in various aspects. So here he was talking about it's up to us to take our communities in our hands and make a difference. And he held the glue was symbolic. He was fixing the crack in the wall, or the cracks in the communities. He came from a community that's fairly marginalized, but he wanted to show the good things about it. Okay, as you all know, and we've been discussing, the area of research on place effects on health is burgeoning. And we need a way to make sense of this evidence. And I'm a systematic reviewer, that's one of my hats. And we try to bring together evidence in a coherent way that minimizes bias, so that people don't have to read, policymakers and practitioners, people in the planners, they don't have to read all of the various studies, they just have to read one brilliant one. What? No, what? Anyway, it's a good way of bringing disparate pieces of evidence together and trying to make sense of it. You'll see it's kind of hard with this body of research. So right now there are at least 15 reviews. Some of them are systematic, and at least one of them Larry was involved in, a very good one with Jacqueline Kerr, on studies of the neighborhood environment and adult health. 4 of these are for older adults only. So then you ask, why do we need another one? Why yet another systematic review? Well, most of the reviews mixed adults of all ages. The existing reviews combined cross-sectional and longitudinal. And that's because it's hard to find a longitudinal study. At least 2/3 of the evidence is cross-sectional, and only 1/3 or less is longitudinal. And there's also a need to study older adults separately to really understand how the environment impacts on them for all the reasons we've discussed before. And I wanted to review longitudinal work only because, as we know, longitudinal can help us get at causation. Okay, so what we wanted to do was conduct a systematic review of longitudinal studies in higher income countries, higher and middle income countries. And we have a number of specific questions. How does place affect the health of middle-aged and older people over time? What are the mechanisms through which it acts? What are the methodologies and the methodological challenges? And there are quite a few. And what are research needs going forward? Okay, so this is the conceptual model that I used for kind of orienting the review. And the one thing about conceptual models and reviews, they change as you go through, as you find out more. And this model was based on the one in Kerr, the one that Susan mentioned from the Systematic Review of Mobility, and also some theoretical literature I was reading. So it shows that basically there's the larger environment, the neighborhood environment, which consists of the natural environment, the built environment, and the social environment. And like Larry, I'm particularly interested in the natural environment, and this is one that hasn't been studied much. There are a couple of pathways through which the natural environment can affect health. It can, one, reduce stress. You kind of go into a place of beauty and you go, this is wonderful. It can also help to focus attention, and quite a few studies, all with university students, have shown that if they exercise in natural places, their stress level goes down compared to going for a run on a city street. They can also provide amazing places for creative physical activity. And so the stress versus rejuvenation, and then the health behaviors. I won't go into all these pathways, but the important thing is it's complex, and it's hard to know where to put the mediators or moderators, because we can see physical activity as moderating the effect of physical environment on physical functioning, but we also, as Abby pointed out, might see physical functioning as moderating physical activity. Okay, so what did we do? Well, we included only longitudinal studies, and this is where it gets complicated. They had to have measures at the neighborhood level and at the individual level. And so it gets a bit tricky and a bit frustrating, because what about someone's perceptions of their neighborhood? Well, we said someone's perceptions don't count if they're not aggregated up to some unit. So a neighborhood, a census tract, something like that. There had to be a measure, a consideration of a neighborhood or a place, whether it's the 1 kilometer buffer or whatever. And they had to have at least 6 months follow-up. Participants had to be age 40 and up, and they had to be a population sample, not a clinical sample, not like schizophrenics. And they had to have health outcomes, physical, mental, or social health, or health behavior. So this poor review has been around since 2011. It started off life as just a small aspiration to review all the literature on the place and health, on longitudinal studies of place and health for every age. Well, we tried to do that. It was funded by a CIHR grant to Ron Labonte on globalization and the health of Canadians. We tried to do that. It didn't work so well. It was way too messy and way too complicated. So the first 2 searches are from that. And then the second search is on all ages only. And I've also searched reference lists of included reviews and studies, and we will be doing a broader search, because I'm not convinced we've gotten everything, and you will probably tell me that we haven't. Just to minimize bias, 2 people did everything and checked on each other's work. So what have we found? Well, we had 5,000, almost 6,000 articles from electronic searches, and 37 were included from the electronic search. Now, this is why I think we haven't found everything, because just looking at reference lists from reviews, I found 7 that were missed in the electronic search. So this is why we'll be doing the electronic search again. Now, 44 studies are included. 4 are from Sweden, 3 are from England, 1 is from Italy, all the rest are from the USA. Not much from Canada in terms of longitudinal. Now, I know we're doing great work, but so far I haven't found any longitudinal studies. So now let's get to the results. This is for chronic disease and mortality. There was a lot on it, but it wasn't very satisfying, because most of it used SES as the indicator of place. So socioeconomic status of the census tract, of the neighborhood. It was, in general, there was a trend towards more positive studies, so i.e. people who lived in neighborhoods with lower SES had lower mortality, lower incidence of CHD, lower incidence of progressive kidney disease. The effect sizes were small to moderate, kind of 1.5 to 2.5 in terms of hazard ratios. One study did look at something a bit more interesting, population density. And they found that people had higher mortality with greater population density. Cancer, progressive kidney disease. So lower SES was in general linked to most conditions. But that doesn't tell us much, does it? What does it? We know that there's something going on, but we don't know why or how. Now, we get to a few more interesting studies, and you'll notice I spent a bit more time on these. One nice study that was done in Portland looked at neighborhood walkability. And low neighborhood walkability predicted hypertension, flat out. But what was interesting is that the food environment was only important in low-income neighborhoods, in low walkability neighborhoods, sorry. So there was an interaction between the food environment and the physical environment. Diabetes, if people perceived that there were better resources for physical activity, more recreation facilities, nice walkways, more parks, and for healthy eating, they were less likely to have type 2 diabetes. So mental health and cognition. So there are a few interesting pieces of information. Mental health and cognition so far is pretty well a washout as far as environmental place effects from longitudinal studies. Now, I have several theories of why, but basically for SES it was equivocal. 2 found yes, there's some effects, 2 found no. Social fragmentation or cohesion is hypothesized to be highly related to mood, and you would expect so if you live in a neighborhood with high traffic, high crime, lots of litter, graffiti, then you're more likely to be stressed all the time. But that hasn't panned out. Poor aesthetics, again, relating to this idea of beauty, it was unrelated. And the crime rates or the perceptions of violence were unrelated. But people haven't looked at the natural environment, and people haven't looked much at walkability. But depression is an interesting thing. It's kind of not always, it kind of fluctuates. So that might be another issue with studying depression. You have to be very careful of the time period with all of these outcomes. So cognitive change. 2 pretty interesting studies. One, higher perceived disorder, things like litter, graffiti, broken sidewalks. They were taken from a separate sample. So they went and asked a separate group of how they perceive their environment. And the perceptions of poor neighborhoods at baseline were related to greater cognitive decline. So if there were poorer environments, there were greater cognitive decline. Now, interestingly for Mike, the ApoE4 allele was only important for those in low disorder neighborhoods. The theory is that when there's high disorder, high fragmentation, high crime, that nothing matters. That is responsible for most of the effects. But when there's low disorder, then the genes can express themselves. And another found that living in low socioeconomic status neighborhoods was related to greater cognitive decline. So these are kind of intriguing. There's probably something there with cognitive decline. Now, incident mobility disability. These were the studies that I found the most intriguing because they really tried to get at mechanisms. So 3 studies, higher number of observed and perceived poor neighborhood conditions, things like the poor housing, poor condition of sidewalks and yard, noise, air pollution, poor street conditions, potholes, was related to a higher risk of incident disability. And the relative risk was pretty high, 3 times as high in this study. So people who lived in neighborhoods with social fragmentation and disorder were 3 times more likely to develop functional limitations. Older adults in car-friendly neighborhoods, neighborhoods where most people drove to work, were more likely to develop functional disability. One study in England, I don't know if you guys know, but they have this wonderful index of multiple deprivation. The British are very concerned with socioeconomic differentials in health. This index measures all kinds of things. It measures health, it measures the built environment, the social environment, socioeconomic conditions, housing. But the problem is, for research and for really trying to untangle, they put it all together in one big index. And the researchers said they can't disaggregate this. So it's kind of frustrating. Yes, it's related, but what part of it? Is it the fact that there's a built environment? Is it the neighborhood social cohesion? Is it the poor housing that's related to the disability? So basically, if people lived in these neighborhoods with multiple deprivation, in the highest quintiles, the highest multiple deprivation, were more likely to develop gait impairments, objectively measured gait impairments. And in another one, perceived neighborhood safety was related to greater likelihood of incident functional impairment only for people in low SES neighborhoods. That's perceived neighborhood safety. The crime rate, the actual observed crime rate, was not significant. So in some cases, perceived safety really affects behaviors and physical activity. And again, this is a photo voice picture. We're getting onto the health behavior aspect of it. This woman wanted to show the wonderful facilities in her neighborhood available for physical activity. So health behaviors. Again, you'll see a pattern. You'll see why I get frustrated, because there's one study here, one study there. They all use different measures. They all use different outcomes. It's hard to bring them together. So in 3 studies, walkability or access to resources was related to lower increase in BMI. So those that lived in neighborhoods with poor access to resources, poorer walkability, were more likely to gain weight. And I think it was something like 1.5 pounds over a few years. And they also showed less, people in these better resourced areas showed less decline in walking. But in one of them, it was only important for high SES men. So you can see why it gets frustrating and why it's complex, because in some, walkability is important for low SES men, in others it's important for high. And fast food accessibility, again, was only related to BMI in low walkability neighborhoods. In one study, greater urban sprawl was related to higher BMI and to less walking in cross-sectional. But that effect completely disappeared when they studied it longitudinally. So that kind of shows why we need longitudinal work, because people self-select into neighborhoods. There's a self-reporting bias. So what do I conclude? Well, I conclude that there are a lot of limitations with the work, that it is the most frustrating systematic review I've ever worked on, because it's not nice and neat. It maybe is trying to cover too much, but every study uses different measures of walkability, of social cohesion, of deprivation, of resource availability, and there's got to be some consensus somewhere. So it's really hard to try to figure out how to meta-analyze these. And every study uses different outcome measures. So some limitations to this work were that there's the same source bias. Some studies assessing the perceptions asked the people themselves what their neighborhood was like. And you can see that depending on personality, depending on motivation, that might color their view of how walkable, how safe the neighborhood is. So we really need those studies that bring the perceptions up to the neighborhood level and where those perceptions are drawn from another sample. And several studies, and as a measurement person, this drives me nuts. Everybody loves indices. People are really interested in indices. All the time I'm getting asked in Ottawa, why don't you create this index? Why don't you create that? But several of the indices lump the physical and social environment together. And we really need to understand how they act separately and how they interact. Many studies have self-reported outcomes. Now that's important, but we also need some of these measured outcomes, some of these objective outcomes. And as you've seen, there are only a few studies that move beyond SES and social environment. They are coming, and the evidence for mortality is pretty clear that SES is related to mortality and to chronic disease, but what else in the built environment is? It seems like social disorder is related to functional limitation, and neighborhood resources are related to walking and to BMI. But not much has been found so far for depression. And I don't know that that necessarily means there's nothing there. Okay, so what do we need? We need not just more studies. Of course we need more longitudinal work. As I said, I only found 44. There's probably others out there, but there's more longitudinal work. But we have to have more thoughtful work. Work that really is based on theory, on mechanisms, and testing those theories and the hypothesis about those theories. We need to pay attention to the mediators and the moderators. We need to have more focus on the built environment, objectively measured. Perceptions are important, and in some cases, as I've shown, they're more important for actual behavior, but we also need to know what that objective aspect is. We also need studies that focus on the built and natural environment measured separately and their interaction. And as Larry said, and as I kind of showed, I'm really interested in this idea of the natural environment. There has been very little, if any, longitudinal work done, except for one. Oh, I forgot to mention the Japanese study. There was this nice Japanese study that wasn't included, where they asked people in Japan about how nice their neighborhood was for walking. Were there a lot of trees? Could they walk to a lot of parks? And it was related to mortality 7 to 8 years later. So that is one of the only hints that we have that there's something there. But evidence from short-term studies shows that, yes, the natural environment does impact our mood and our physical exercise. And we need to understand how the interplay between objective and perceived measures, because, as I said, both are important. And studies should also consider time in neighborhood. If someone just moved into a neighborhood, it's going to have less effects on his health than someone who has lived there for 10 years. But some studies actually limit it to long-term residents, others don't. Studies also need to consider people who move in and out of neighborhoods. And we also need some well-crafted intervention studies, again, based on theory and based on evidence. Okay, that's it.
So any questions for Betsy?
I'd just like to say I really appreciated your summary of that. I think it's just really right on. And I hope there's a way for us to tackle some of that for the next day and a half. Some of those things.
Thank you.
You had a comment, the instrument, what was the bias? What was the same people?
Same source bias.
I didn't understand. I understood the best, but I didn't understand how the mechanism there.
Basically, same source bias is that if you ask people about their environment, they might be colored by their motivations, they might be colored by their personality. If someone's depressed, he might report his environment as poorer. He might be more likely to see what's wrong with the environment. Whereas someone who's an optimist like me might think, the worst street is wonderful. So we really have to get the perceptions from people who aren't responsible for the outcomes.
So you want other people, like your
I know.
Not your participants, but other people in the neighborhood would give you the perception.
That's right.
Of the neighborhood.
That's right.
They have their bias.
I know, but they aren't the ones that are saying, I walk. See, someone who doesn't walk a lot might say, I don't walk. Yeah, it's not as systematic. So it might be random.